All right, guys, how's it going? Okay, I'm going to try to run through 8 here, which is a super complex one here. So I'm sure we have to refer to my book a couple of times, but this is a great chapter. I actually read through it a couple times. I already made your short answer questions out there up, and we're going to talk about those this week in class. But it is pretty intense. It's got involved a lot of cuss words called chemistry. So I like chemistry in it. Okay, let's take a look. I'm going to share my screen, and we're going to pull this up. Okay, and we're going to dive on into it. Oh, share. I didn't hit it. Okay, you should be good to go there. All right. Oh, now it's not letting me do what I need to do. All right, here we go. So break down the chapter, overview of cell respiration, then what happens outside the mitochondria with glycolysis, what happens inside the mitochondria with fermentation, outside the mitochondria fermentation, and then inside the mitochondria, we got two big events. Now, basically, one of the things I want you to think about here is there are 5 processes we're looking at, or 5 metabolic pathways, okay? In the cytoplasm, we have glycolysis and fermentation or anaerobic cellular respiration. And then inside the mitochondria, we've got this preparatory reaction that's just one reaction that sets up the Calvin-Benson, I'm thinking about those plants. The citric acid cycle, also called the Krebs cycle, because Dr. Krebs discovered it. Okay, and then we're going to have the electron transport chain where the most ATP anywhere is made, 32 ATPs. That occurs in the mitochondria on the inner membrane called the crista. Okay, so we're going to dive into it, and then we end the chapter with a little discussion of metabolism. Okay, so we'll talk about that. All right, so first off the bat here, right, in cell respiration, we're going to do what? We're going to break down the sugar molecule, which has potential energy, and we're going to use oxygen to be the final electron acceptor, which is one of my favorite test questions. Oxygen is the final electron acceptor in aerobic cell respiration, and we're going to release that energy, power in the bonds, and we're going to use that energy to pump hydrogen across the membrane, electron transport chain, and to make a pile of ATPs. Some players we're going to see include electron transfer molecules, NAD and FAD, that are very similar to NADP that we saw in the chloroplast to carry electrons from the light-dependent reaction to the Calvin-Benson cycle. But here, they're going to carry electrons to the electron transport chain, as we'll see. All right, I forgot in the last chapter, but don't forget, you got to write down the equation for photosynthesis. Well, here's the equation for cellular respiration, okay? So C6H12O6 glucose is going to be oxidized into carbon dioxide, 6 carbon dioxides. In doing that, we're going to use 6 oxygens, and they're going to be reduced because they're going to receive the electrons from the sugar molecule, and they're going to be forming water from that because there's plenty of hydrogens around. Now, if you'll look at this, the carbons are equal on both sides, the hydrogens are equal on both sides, as is the oxygen on both sides. So remember, first law of thermodynamics, you cannot create or store anything, you can just rearrange things, and that's what we're doing. And in doing that, we're going to make 6 CO2, 6 waters, and potentially 38 to 36 to 38 potential ATPs. So electrons then are moved from the substrates and received by oxygen, and then those there is oxidized and oxygen is reduced. Okay, I think we beat that horse in the ground now. So one more time, in the cell of the cheetah, right, or whatever that is, okay, and that mitochondria is where all this is going to happen. We're going to make all that energy, and we're going to make some CO2 and water. Matter of fact, one of the coolest things about this is these two gases are what we exchange every time we breathe in and out. So the oxygen we breathe in is put into our bloodstream and our lungs to do this, to be used right here in the mitochondria, in the electron transport chain, to be the final electron acceptor, so we can make all these ATPs right here on that crystal. And then the CO2 that's made here is what we breathe out. It leaves our blood and goes into our alveoli and is breathed out. So this is super cool to be, every time, that is those two gases are directly related to cellular respiration. Okay? Now we already mentioned nicotinamide adenine dinucleotide, NAD, and FAD are 2 coenzymes that are involved in cellular respiration. NAD is going to carry higher, and they're electron transfer molecules. FAD, also electron transfer molecule, and they're going to carry those electrons from all of our reactions to the ETS, so the high energy electrons can be used there to make the ATP. They are recycled, okay? And the niacin vitamin is the N, and riboflavin is used to make FAD. So these are 2 super important molecules made from vitamins that are totally required for cellular respiration to occur. Okay, and they are energy transfer molecules. I almost think of them as someone like the gas company. You know, you get if you got propane in your house, right? You go pay, they fill up the tank, and they go to your house and dump it, and they go back to the shop and get more. And these guys go back and forth carrying these energy, these electrons, as we'll see. Okay. Now, just to kind of review what I've already said, we're going to look at glycolysis, which is a metabolic pathway occurs in the cytoplasm of all living organisms on Earth. Then we're going to talk about fermentation, which is doesn't require oxygen. That also is in the cytoplasm, which I want you to know where these occur. Real big on these 5 processes: what goes in, what comes out, and where they occur. Okay. And this preparatory reaction, it's just one reaction that occurs in the mitochondria. Then we're going to see the citric acid cycle, also called the Krebs cycle, where we're going to make a truckload of NADPHs and make some carbon, make a bunch of carbon dioxide and some FADHs, and those then will then be used in the ETS here to make 32 to 34 ATPs. So our 5 processes then would be glycolysis and fermentation in the cytoplasm, preparatory reaction, citric acid cycle, and electron transport chain in the mitochondria. Okay, and so here we go. We can see it here. Then the sugar is going to come in, right? We're going to eat it from a plant, or they use it to send their mitochondria, right? And we're going to do glycolysis and see a sugar called pyruvate being made. And doing that 10-step reaction, we're going to end up using 2 ATPs, and you end up making 4. So you end up getting a net of 2. So if you pay $2 for a bicycle and you sell it for 4, how much money did you make? You made $2, right? And small organisms can live off of that. So fermentation is the way to reboot those NADHs, which have potential energy in them that were also made here, back so we can recycle that. And we'll see that later. And then that preparatory reaction is in that mitochondria with the citric acid cycle and ETS. Okay, there it is, preparatory reaction. And then citric acid cycles occurring in the mitochondria, and then ETS, where we're going to make 34 ATPs. So you can see a lot of electrons are being moved here, and it all comes to tuition there, where we have a total of 2 ATPs until we do ETS. We have those 2 there, right? And we actually make 2 in the citric acid, so we made 4 ATPs, and then we turn around and make Shazam, 32 or 34 in ETS. Okay. Now, the thing to remember is to not have a nervous breakdown here, okay, first of all, and to keep breathing, okay, and just step away from it and come back to it. It took me several college classes before I got a feel for this, okay, and I think I have a pretty good feel for this, okay? So glycolysis then is 10 reactions that occur in the cytoplasm of just about every cell on Earth, okay? And so this is one of those universal first steps to breaking down sugar to get energy, ATP out of it, okay? So we're going to have investment step where you put 2 ATPs to break the sugar apart, and then the harvest reactions where we're going to end up making 4 ATPs. And we're going to make them a little bit different than we made them in the chloroplast back. Well, actually, it's not going to be that chemoosmotic where the hydrogen are pumped. This is an enzyme that grabs this substrate level phosphorylation enzyme, just takes an ADP and slaps a phosphate on it from the energy in the molecules of the sugars being produced, okay? And we'll make 2 ATPs that way. So in glycolysis, we will put 2 in, we will net 4, and you'll end up, so look what you need. So you have to have the glucose, 6-carbon sugar. You need 2 NADs because they're going to carry the electrons off that sugar, remember, power in the bonds, and you need 2 ATPs, okay? And of course, but I guess you need the ADP there, but you make 2, you put 2 in, and then you net 2 3-carbon sugars called pyruvate, 2 NADHs, which is stored energy from that sugar molecule now on the NAD, it's reduced. And then you've made 4 ATPs, right? And so for a net of 2, okay? Very cool, all right? So here we go, we're gonna look at it real quick. So here's glucose. And everywhere, each of those, there's 10 enzymes that catalyze this reaction. So glucose is gonna be converted into, bam, let's see what we do. So glucose is 2 ATPs are put in, and you end up with 3-phosphoglycerate, okay? And then you do another enzyme here and use 2 more ATPs, right? I think it's that right. I think I'm getting that wrong. So it comes in first and you use 2 ATPs there, okay? And I think it's still looking at that. And then it goes down, and that's all we use, right? Okay, and then you end up breaking those into 3-phosphoglycerates, and now it separates, and you use 2 more ATPs. Actually, let me get my story straight here. We burn another. Actually, we don't. That's just confusing to me. Okay, bad pictures here. Let's start over. Sorry. Okay, so you use 2, and all that's still at the top. That's still right there. So we split it here, and then the three phosphoglycerates then are going to have energy in them that's going to be ripped off, and the enzymes are put on the NAD to make NADH. So you end up making 2 NADHs here, and you end up with this 1,3-biphosphoglycerate. Okay, so we're converting the glyceraldehyde 3-phosphates into 1,3-biphosphoglycerates. And there are enzymes. There we see a red arrow. There's an enzyme involved. Okay, or a black arrow. Black arrows are reactions. One, another enzyme does this, and then another enzyme does this. Okay, and then it's still showing that last part. Then look what happens. We're going to come in and we're going to make an ATP here on each side. So now we put 2 in, and we've now made 2. And this is that substrate level phosphorylation where enzyme takes a phosphate on the ADP from the energy as that molecule is converted from BP3, 1,3-biphosphoglycerate to the 3-PG, 3-phosphoglycerate. Okay, very wild. And then we're going to do it again and get another ATP out of there, I believe is coming up. Okay, hang on. Yep, that's another enzyme there, another enzyme reaction. And here it comes. Finally, we do it again down here. So we've got 1, 2, 3, 4, 5, 6 of the reaction. There's 10 in there. We're missing a couple of them. There's enzymes there, 4 enzymes. Okay, and so we end up making 2 more ATPs there. So there it is, kind of laid out right, and you end up with a net of 1, 2, 3, 4 ATPs. Put 2 in for a total net of 2. And the 2 pyruvates, 2 3-carbon sugars. So all we've done is we cut that sugar molecule into 2 3-carbon sugars, and the energy from that is now used to make 4 ATPs, but we put 2 to get it kick-started, the energy required. So you have a net of 2, and the energy left in those 2 NADHs is going to be used later to make ATP in the electron transport chain. Okay, pretty fascinating. Sorry, I didn't go through the step art, and they kept repeating themselves. So there's your summary of it right there. Okay, here is that phosphate level phosphorylation where the substrate level ATP synthesis where the enzyme grabs that phosphate off of the BPG and it becomes 3-phosphoglycerate, and that energy is put on the ADP to make the phosphates put there to make ATP. So we pull a phosphate off of that to make the ATP. Pruvate then is potential energy, and it is going to be taken into the mitochondria, and the transition reaction or prep reaction is going to affect it, and then it gets put into the Krebs cycle or citric acid cycle, and we make all these NADPHs, more NADHs, and then the FADH2s, and those are going to be used to make energy level later. However, if oxygen is not available and we can't, because the mitochondria requires oxygen, then we can fermentate or do fermentation or anaerobic respiration to reboot this so that we can break down more glucose, convert it to pyruvate to make more ATP. If oxygen is available and you have a mitochondria, then we go over to into that and do the other steps, do the prep reaction, Krebs cycle, also called citric acid cycle, and ETS. Here is how fermentation is. It's an anaerobic process that reduces pyruvate to either lactate or alcohol and carbon dioxide. The energy in that NADH is transferred to the pyruvate, and then pyruvate is converted into alcohol or lactic acid or alcohol and carbon dioxide or other compounds. Okay, this is how we make alcohol spirits and bread. Those big fat yeast rows, it is the CO2 leaving that causes the bread to rise. It is the CO2 that gives beer its fermentation and causes the carbonation that we see in some of the wines, etc. Okay, lactic look here. Other cool stuff: lactic acid fermentation carried out in certain bacteria and fungi produce lactic acid, used commercially to make cheese, yogurt, sauerkraut. Other bacteria produce chemicals anaerobically, including isopropanol, propionic acid, and acetic acid. So these are pretty cool. Here's the equation for fermentation. Notice this is all this to here is glycolysis. Okay, but we need more NADs, right? So the Na2 NADHs, then the energy and hydrogens come off of the NADPA or NADH, and they're put on the pyruvate to either make alcohol and carbon dioxide, that's carbonation, or 2-lactic acid, which is another sugar that can later be converted back into glucose and carried through the prep reaction, and Calvin, excuse me, and Krebs cycle, citric acid cycle, and ETS. Now, here's the deal. When you do this, you recycle and you get 2 NADs. You can go back and get another sugar, another Pruvate, and take this through. But each spin of this, it just makes 2 ATPs. But if you're a single cell organism, you can live off of fermentation, right? Because you can reroute your NADs to break another sugar down, right? And you've got still energy, because if you drink a lot of alcohol, you get fat. So there's energy in alcohol, and there's still energy in that lactic acid. Now, this process of the actual 2 ATPs represents about 4% of the energy in a compound, 3.9, something like that. So this is a low energy yield. We use this in exercise when we are already running the other system as fast as we can, and we can't go any faster, or we're running low on oxygen. And this gives us that burst of energy in the fourth quarter, but this also means you usually pass out after that, or you're wore out, okay? Because we say you fermented, you went into fermentation, and that causes burnout real quick. So an athlete, the better shape you're in, the less likely you got to do fermentation, the longer you can last doing a strenuous exercise. And these marathoners, they can go for hours because they don't fermentate. They're so well-oiled machine that they can do glycolysis and then do that prep reaction and the Krebs cycle, also called, the citric acid cycle, and then ETS, where the oxygen's used and all the ATP's made, and they're like a little machine. So I always think of marathoners or triathletes as little machines who are running like a fine oil machine, okay? So fermentation, also anaerobic respiration, pretty cool there. Advantages, you can get a quick burst of ATP for muscular activity that I just said. But the disadvantage is you could be, but you could have, you could be wore out after that. You could have lactic acid, alcohol are toxic to cells, so that's not necessarily good. The lactic acid changes pH and can cause muscles to fatigue. You could have oxygen debt where you need more oxygen, right? And really it burns up little electrolytes and stuff in there. Yeast die from the alcohol they produce for fermentation. The efficiency is 2 ATPs. Oh, and I said it was 3.9. I'm a liar. It's 2.1. So I missed that one. But really low number. So that's pretty good. 2.1% of the total energy in glucose is converted to ATP there. So that's bad. Okay, compared to 3638, which is 39% efficiency. Okay, and there's our little equation showing input glucose and the 2 ATPs that were made, right? And glycolysis, and then you convert the glucose, and those NADH is used to make the lactic acid and our alcohol and carbon dioxide, and those NADs now are free to go back and grab more electrons off another sugar molecule. Should have put that in that picture. So you end up with the same net of 2, but you now can reboot the system again and can do another glycolysis and convert that another Pruvate into lactic acid or alcohol and CO2. Okay, so that's pretty confusing, right? But that's 2 of our pathways. And now on normal cellular respiration now, carbon dioxide, the Pruvate, the 3-carbon sugar, the 2 pyruvic acids are Pruvate, same thing. Pruvate is pyruvic acid. They now are going to go into the crista of the mitochondria, into that open space. The crystal being the membrane, and the space in there, inside of that area, it would be, I guess, the equivalent of cytoplasm, right? But it's in that inner space that this occurs. So look what happens. Pruvate is converted to 2-carbon acyl group that we call acyl coenzyme A, and the electrons are pulled off of Pruvate and hydrogenized laying around to reduce NAD to NADH. And we make a carbon dioxide. So what you're doing here, think about it. You've got 6 carbons. We now cut it into 3, and now each of the 3 are going to have one of their carbons pulled off. So now you're going to be left with a 2-carbon acyl coenzyme A, and you've made a carbon dioxide, and you have another NADH. And since you had 2 pyruvates, it's got to happen twice. So the products here would be, take a look, you end up with 2 pyruvates, and you end up breaking them down, and you make 2 carbon dioxides right there, 2 acyl coenzyme A's, and 2 NADHs, which represent potential energy that can be used to make 3 ATPs apiece in the electron transport chain, or a total of 6. Okay, so that's just one reaction. But what we're doing now is pulling carbons off of our sugar, right? We first had 6 carbons, we tore it apart. Now we have done what? Pulled a carbon off each of them and made our first 2 CO2s. Keep that in mind. It's kind of cool. Okay, so this occurs in that matrix area. I couldn't remember the term a while ago. So the matrix is the fluid part. I still remember the great movie, The Matrix, right? And the folds are called crista. So that transition reaction occurs in that matrix. And now we're going to see also in the matrix the Calvin, I'm wanting to do photosynthesis, sorry, not the Calvin-Benson cycle. That was building the sugar. We're going to now see the Krebs cycle, also called the citric acid cycle. Okay. Now, here's just some notes, and I've already said all this. It's also called the Krebs cycle, occurs in the matrix of the mitochondria, and is a circular metabolic pathway where the final, you actually have citric acid in there, and that's where it gets its name, citric acid cycle. We're going to make, listen to the products. It sounds like I always do this to the rhyme. On the 12th day of Christmas, what you love gave to me, 4 carbon dioxides, 2 ATP, 6 NADPHs, 2 FADH, and a partridge in a pear tree. Those are your products, sorry. 4 carbon dioxides, 2 ATP, 6 NADHs, and 2 FADH2s. Now, the difference between these two is, think about this. They're carrying electrons. They're almost kind of remind me of rechargeable batteries. NAD would be a AA, bigger battery. FAD would be a AAA battery. So it can't carry as high of energy electrons, okay? And that's going to be kind of cool in a minute. We'll see that's why. So it's a lower energy electron it's carrying, okay? All right, let's take a look at it. Take a deep breath, and here we go. So here it is. So silicoenzyme A comes in. Here it is. We've made our silicoenzyme A. It binds to a 4-carbon sugar here and forms citrate. Citrate is converted to isocitrate, I believe. They didn't give me the intermediates here. Then it forms another intermediate, another intermediate, another intermediate, and then it reboots itself back into the other intermediate here. And I thought they might have given us those. I always like the names. You see, it's been a while since I've had those. It's kind of irritating. They could have mentioned those. Kind of shocked if they didn't. I'm looking over here to see why they didn't list those. Kind of shocker. Okay, but you have to draw those out in major biology. But those are pretty cool. I believe one of them is maltase, right? That sound in malt. Okay, but anyway, now but look at the punchline. So it's not important that you know those intermediates because we didn't tell you it's not to know them anyway. But it's 1, 2, 3, 4, and then it reboots itself. Okay, but citrate being the first one made does citric acid cycle. Okay, now check it out. We made 1, 2 NAD, 3 NADHs, 1 ATP by substrate level phosphorylation, and 1 FADH. But since we had 2 pyruvates, this has got to, and 2 acetylcoenzyme A, it's got to spin twice. So you get this total here of, and I didn't count carbon dioxides, 1 carbon dioxide, 2, 3 NADHs, 1 ATP, 1 FADH on 1 spin. But when you spin it twice, you get 4 CO2s, 2 ATPs, 6 NADHs, and 2 FADH2s. Okay, maybe they're going to show it to us here. They still ain't showing me the intermediates, okay? But you can see it again. They're going to show them coming off. And now, what happened to our 2 carbon acetylcoenzyme A? It's gone. So all 6 carbon dioxides have been made when this runs twice, right? So we made 4 carbon dioxides. We made 2 in the prep reaction. And so we've got the sugar's gone. We haven't used oxygen, but we now have all the carbon dioxides that we see in our equation for cellular respiration. Okay, and now she's just kind of walking through that again. Right now, watch what's going to happen now. So far we had 2 acetylcoenzyme A's go in. We needed 6 NADs, so we made 6 NADHs, 2 FADHs, and 2 ATPs, right? And now all these NADHs, the FADHs made here and the prep reaction and in the glycolysis are going to be carried to the electron transport chain, where you have this complex array of proteins and what are called cytochromes that form this electron transport chain that's on the crista of the mitochondria that is going to allow us to make lots of ATP. It's on the plasma membrane of some aerobic prokaryotes. That's kind of cool, okay? And we're going to see also the use of oxygen here, okay? So hydrogens, right, from NADH are going to be delivered here, and they're going to be pumped into this matrix, and this into this outer membrane space between the cis crista and the cell membrane, the outer membrane of the mitochondria, as we'll see in just a second. Check it out. We have another electron transport chain like we saw in the chloroplast. Let's get to the picture of it. Here it is. So here is the electron transport chain, right? This kind of looks like the chloroplast, but there are no photosystems here. And look, NAD gives up an electron, high energy electron. It is transported to check it out here. Coenzyme, what is that? Coenzyme Q. Then it goes to cytochrome reductase over here. Then it goes to cytochrome C, and then cytochrome oxidase. Now, that energy of that electron, every time it goes from 1, 2, 3, 4 different components here, 5 in the whole system, then that electron is worn out. And as that occurs, the energy is used to pump hydrogens into this outer space. See the hydrogens going to outer space, into the outer space, okay? Now, when the electron has no energy left, half an oxygen molecule, and O grabs a hydrogen and forms water. So oxygen is the final electron acceptor for this low energy electron in the cytochrome oxidase, the last reaction. Then all these hydrogens build up, and they're going to go through the ATP synthase like we saw before. Instead of this time, it's going into the membrane instead of out of the granum in the chloroplast, and we're going to make a truckload of ATPs. Matter of fact, for every NADH, we make 3 ATP. Enough hydrogens are pumped to make 3 ATPs here by chemoosmotic ATP production. That turbine being turned as hydrogens go through, slapping a phosphate on to ADP. And FADH skips the first pump, gives up its electrons to cytochrome Q, and you get enough hydrogens pumped to make 2 ATPs, which is kind of cool. So 2 ATPs are made there in the for every FADH. Now in class, I'm going to show y'all a video of this kind of animation. I think it's good. So the net yield then from glycolysis is 2 ATPs, from citric acid plus 2, and from the electron transport chain there's 32. That represents, or up to 32, so up to 34, so 36 ATPs represents 39% efficiency. The rest is lost as heat pretty much, and the energy left in carbon dioxide. Here's a summary showing the ATPs again, right? And look at all that. So 2 NADHs from glycolysis, 2 from the prep reaction, and 6 from the citric acid cycle, Krebs cycle, make all that. And for the odd number 2 comes time, sometimes the 2 from out in the cytoplasm from the glycolysis can't make it. Is given to FAD, right? They end up missing that first pump, and so you only make 2 instead of 3 ATPs on that one. So that's where the extra 2 come from. So if they get carried in there and they get to the first pump, but because sometimes they're giving up to FAD and they skip that pump, some organisms get 4. Now, one of the coolest things about this too is this yield of 3638 is theoretical. And I started teaching major biology 7 years ago, and the other book was a little more detailed, had a lot more rabbit holes than this one, which I like. This is just core stuff you need to know in here, not a lot of rabbit holes. You know, I mean, random, going off on different directions. But one of their little commentaries said that that's never been measured. The highest ever measured in a living cell is 30. So we think there's a lot of leakage going on here, a lot of, the hydrogens leak out of the membrane, whatever. But so we now call this theoretical yield, okay? And as was reported when these guys won the Nobel Prize that did this. But it's funny that only 30 ATPs have ever been recorded in the laboratory. So that was a shock to me. That would be kind of like for us that go to church, I do too, that they found 5 more commandments. That's how shocking that is to a biologist, because the 3638 was, I thought, written in stone like the 10 Commandments, okay? Kind of wild. Okay, now I have a question, a short answer question. I kind of break it down, but food is broken down by catabolism, and then we use it to build our body by anabolism reactions to build things up. So I had this nightmare first time I was teaching this, thinking about this. So we just spent about 20 minutes, 30 minutes going over how we break down sugar, right, to get energy and to make those ATPs. Well, what about the other 2 things we eat to get energy from, proteins and fats? Don't tell me I got another 40 minutes on each of those. Well, guess what? I've got a great answer for you today. Proteins are broken down into 20 amino acids that can be put into glycolysis, transition, citric acid cycle, to then use that energy to make ATP. And all other sugars that we can break down can be put into glycolysis, and all fats can be broken down into 3-carbon glycerols that get stuck into glycolysis, and fatty acids can be converted into acetylcoenzyme A's that can then be used and shot through. So we don't have a metabolic pathway for proteins and fats. We have the main pathway here of glycolysis, preparatory reaction, citric acid cycle, Krebs cycle, and ETS, and that is how we get energy from proteins, fats, and all sugars. Now, sugars like cellulose, we don't have the enzyme to break it down, so we don't get energy from that. That's the roughage in our diet. So this is fascinating. For about 50 bucks, or I could buy a fold out piece of paper and it would cover up the whole wall behind me, and it would have breakdown of all organic molecules on Earth that we can eat, that humans can get energy from. And it would show every sugar, every protein, all 20 amino acids, and all fats that we can break down and get energy from, and all carbohydrates. And it shows where they go in. It's a beautiful thing. Okay, so we don't have another system. This is the master breakdown of food for living organisms for us. Okay. I think it's fascinating. So I kind of asked a little question on that. Okay. And in breaking down those amino acids, we produce a compound that ammonia is going to be taken off. That's called deamination. And that ammonia is in H2, grabs the hydrogen and forms ammonia, which would be deadly. But our liver converts that to urea. And so this may be something you didn't know, but the breakdown of urea, the main purpose of the kidneys is to filter the urea out, right? Nitrogenous waste that's in our urine. And so urine gets its name from it being made from that urea in it. Okay. So think about this. The more protein you eat, the more urea you make. So when you've eaten a steak, the next morning you get up and you have this real orange, yellow, strong smelling urine because you've broken down proteins. And that's kind of cool. So deamination is a way to get that amine group off, and the rest is then carbon chains that can be taken through, as we saw here, either into the fatty acid, either into glycolysis or into the Krebs cycle or as a silicoenzyme A. So pretty cool little side note there about deamination. Okay, and then the old cliche, you are what you eat. We can use that to make our other. We can use those. We can use the metabolic pool of things to make our proteins and fats. We don't get energy out of the. We don't really break down the DNA much, right? So there could be a little energy, but we really don't break it down. Just talking about amino acids and proteins there. And then she kind of ends the chapter going back and looking at how these two are linked together in the flow of energy. So one more time, photosynthesis, right, comes in. The chloroplast, we got the light reaction, making all those NADPHs, right? Okay, and all that ATP, 18 ATPs, 12 NADPHs, and then the light independent, the Calvin cycle fixates the carbon dioxide to sugar. We eat that sugar by glycolysis, right? And then we take it to the mitochondria and make all that ATP, and then CO2 then and water we make then can be used by plants to make more. Fascinating stuff here. I know this is very tough, and I kind of did an overview of it. But I do think I understand this a little bit better than photosynthesis. But once again, this is super fascinating to me and the circle of life here. So when we terraform Mars, we're going to take some plants and we'll start that circle of life and turn it into a habitable place someday, hopefully. All right, I'll see y'all in class this week. I'm going to get chapter 8 up here in a few minutes, so y'all have that available to watch next week. All right, I'll send an email out Monday to remind y'all that's up, so y'all can watch that before class on Tuesday and Thursday. All right, guys, I'll see y'all later. Have a great evening.